Bismillahirrahmanirrahim. I am Dr. Irfan Ahmed Mughal, Professor of Anatomy at Independent Medical College, Faisalabad. Today we will start our lectures, online lectures. Now the first lecture is on first week of development. So my dear students, you know that sexual reproduction occurs when female and male gametes, means oocyte and spermatozoa, respectively unite at fertilization. So gametogenesis implies a specialized process of cell division, meiosis, which uniquely distributes chromosome among gametes. So what is a chromosome? A single chromosome consists of two characteristic regions called the arms, which are separated by centromere. So these are the two arms and it is united with the centromere. So in meiosis, which is a specialized process of cell division that occurs only in the production of gametes. It consists of two divisions, meiosis 1 and meiosis 2, which result in the formation of the four gametes, each of which contains half the number of chromosomes. So here you can see the process of meiosis, and in gametogenesis, from one cell we get the four cells, but in case of female, one ova is there and we will get only one, the other is the polar bodies. So in the next step we will head towards the fertilization. So what is fertilization? It is actually the union of the male and female gametes, and it occurs in the ampulla of the uterine tube and includes almost three phases. So in the first phase, this is a small description of the fertilization. So in the first phase, there is a passage of the sperm through corona radiata. So it results mainly from the action of the enzyme hyaluronidase. Tubal mucosal enzymes also appear to assist the dispersal. So movements of the tail of the sperm are also important in its penetration of the corona radiata. Number 2 is penetration of zona pellucida. It is the passage of the sperm through the zona pellucida is the important phase in the initiation of fertilization. So different enzymes, they are responsible, which are released from the acrosome, the enzymes esterases, acrosine and neuraminidase. It appears to cause lysis of the zona pellucida, thereby forming a path for the sperm to follow the oocyte. Once the sperm penetrates the zona pellucida, the zona reaction, a change in the properties of the zona pellucida that occurs, and it makes it impermeable to other sperms. Then the next step is, you can see here, the fusion of the plasma membrane of the sperm and oocyte. The plasma or the cell membrane of the oocyte and sperm fuse and break down at the area of fusion. The head and tail of the sperm enter the cytoplasm of the oocyte, but the sperm's plasma membrane remains behind. the next step is very important. Okay, with the fertilization, there is completion of the second meiotic division. And the next is formation of the male pronucleus. So within the cytoplasm of the oocyte, the nucleus of the sperm enlarges to form the male pronucleus and the tail of the sperm degenerate. So the oocyte containing two haploid pronuclei is now called as oocyte. As the pronuclei fuse into a single diploid aggregation of chromosome, the oocyte becomes a zygote. So in this slide, you can see the accumulation of the yolk sac and the activation of the maternal genes, transcription and transformation proteins of the activating factors. So in brief, the fertilization is the union of the haploid oocyte and a haploid sperm to produce a diploid zygote. So I have already explained the capacitation. It is actually the conditioning of the male sperm in the female genital tract. So this is very important as far as the examination is concerned. The SEQs or the MCQs. What is the outcome of the fertilization? So why the fertilization was required? Or you can label it as the significance of the union. So number one, second meiotic division is completed. Number two, diploid number is now restored. So it will give a variation in the human species, and definitely there is a determination of the sex at the time of fertilization. And with that, the cleavage starts. So now this is how the cleavage of the zygote occurs. So from one cell, the cleavage, through the process of cleavage, it will change into 2 cell stage, then the 4 cell, then 8 cell, then morula is there, which is definitely having a 12 to 16 cell stage, you can say it, and it is covered by the zona pellucida, a specialized layer, and each cell is known as blastomere. So number of blastomeres are there, definitely number of blastomeres, and this is morula. It is like the mulberry. ये बिल्कुल शहतूत की तरह है जिसे हम मरोला कहते हैं जी. फिर you can see here in the next stage that when on the 3rd or 4th day, especially the 4th day, this morula, it will enter into the uterine cavity. When it enters into the uterine cavity, it is a free floating ball in the uterine cavity, and the pressure or the fluid which is present outside this morula, it will enter, or it is going to start entering into the morula stage, and it will push the cells, majority of the cells, towards one side of the cell. From here you can see the number of cells are accumulated here and also towards a margin of the cell, so along with the pellucida, zona pellucida, and this is early blastocyst, and this cavity is known as blastocyst cavity. But with the disappearance of this zona pellucida, it becomes the later blastocyst, and it is the blastocyst cavity, and this is accumulation of the cells which is towards one pole of this blastocyst, and this is known as inner cell mass, and these cells which are present towards the periphery of the cell, they are known as the outer cell mass, and the pole towards which the inner cell mass is known as embryonic pole, the other pole is known as abembryonic pole. So the most precious part of this whole human being, start of the human being, is inner cell mass. So it is heavier part, you can consider it as a heavier part or more important, and these are the cells which in future will form the ectoderm, mesoderm, and endoderm. So cleavage consists of repeated mitotic divisions of the zygote, resulting in a rapid increase in the number of cells. These embryonic cells become smaller with each successive cleavage division. So cleavage occurs as the zygote passes along the uterine tube towards the uterus. During cleavage, the zygote is within the zona pellucida, a specialized layer. So division of the zygote into blastomeres begin approximately 20 hours after fertilization. Subsequent cleavage division follow one another, forming progressively smaller blastomeres. After the 9-cell stage, the blastomere change their shape and tightly align themselves against each other to form a compact ball of cells. This phenomenon is known as compaction, and it is probably mediated by the cell surface adhesion glycoproteins. When there are 12 to 32 blastomeres, the developing human is called as the morula. Internal cells of the morula are surrounded by trophoblastic cells. The morula forms approximately 3 days after fertilization as it enters the uterus. So how the blastocyst is formed? Shortly after the morula enters the uterus, approximately 4th day after fertilization, a fluid-filled space, the blastocystic cavity, appears inside the morula. The fluid passes from the uterine cavity through the zona pellucida to form this space. As fluid increases in the blastocystic cavity, it separates the blastomere into two parts. The outer cell layer, the trophoblast, which will give rise to embryonic part of the placenta in future, and a group of centrally located blastomeres, the embryoblast, which will give rise to the embryo. During this stage of development, a blastogenesis, the conceptus is known as blastocyst. The embryoblast now projects into the blastocystic cavity, and trophoblast forms the wall of the blastocyst. After the blastocyst has floated in the uterine secretions, as I have described earlier, for approximately 2 days, the zona pellucida gradually degenerates and disappears. Shedding of the zona pellucida permits the blastocyst to increase rapidly in size, while in the uterus, the embryo derives its nourishment from secretions of the uterine gland. Approximately 6 days after fertilization, the blastocyst attaches to the endometrial epithelium, usually adjacent to the embryonic pole. Definitely it is more precious because in future the whole embryo will develop from this embryonic pole area. As soon as it attaches to the endometrial epithelium, the trophoblast proliferate rapidly and differentiates into two layers: an inner layer of cytotrophoblast, almost having a single nucleated cell, mononucleated cells, and outer layer of syncytiotrophoblast, which consists of multinucleated protoplasmic mass in which no cell boundaries can be observed. At approximately 6 days, the finger-like processes of syncytiotrophoblast extend through the endometrial epithelium and invade the connective tissue. By the end of the first week, the blastocyst is superficially implanted in the compact layer of the endometrium and is deriving its nourishment from the eroded maternal tissues. A highly invasive syncytiotrophoblast expands quickly adjacent to the embryoblast, the area known as the embryonic pole. The syncytiotrophoblast produces enzymes that erode the maternal tissue, enabling the blastocyst to burrow into the endometrium. At approximately 7 days or 7th day, a layer of cells, the hypoblast, which is actually the primary endoderm, appears on the surface of the embryoblast facing the blastocystic cavity. So this how an events immediately following the sperm penetration. So how the pronuclei are formed and then the daughter cells are formed. So this is a brief layout of the lecture. The first cleavage produces 2 daughter cells, then morula is there, then by the 4th or 5th day the pre-embryo consists of 100 or so many cells, the blastocyst. Blastocyst is a fluid-filled hollow sphere composed of single layer of trophoblast and inner cell mass. Now this is the fimbria of the fallopian tube. This whole part is the fallopian tube, and the fertilization occurs in the most dilated part of the fallopian tube, which is known as ampulla. So you can see here the movement and formation of the morula, which occurs in the fallopian tube, and this morula, it will enter into the uterine cavity on 3rd and 4th day of fertilization. So it is a free floating ball in the uterine cavity for 2 days. This you can see here, the secondary side, the penetration, then pronuclei, then the zygote. So first week the whole ball-like structure is known as a zygote. So in the 2-cell stage cleavage starts, and 4-cell stage, then 8-cell, then morula is there, and it is going to enter into the uterine cavity, then the formation of the blastocyst, and this total is known as blastocyst, and this is known as blastocystic cavity, and this is inner cell mass, this is the embryonic pole, this is the abembryonic pole. Then from the cells which are known as the inner cell mass, they will arrange themselves into first 2 layers. The outer layer or epiblast or inner layer is hypoblast. So from here the cells, they move, which is covered by totally by the trophoblastic tissue, which you can divide it into 2 parts. One is cytotrophoblast and syncytiotrophoblast. So here you can see the movement of the syncytiotrophoblast. This is the blastocyst, then the trophoblastic tissue, trophoblastic, how the future placenta is going to form. I will cover it into the next 2 or 3 lectures. This whole lecture, it is taken, or it is based on the 2 books, The Developing Human by Moore and Prasad, and Langman's Medical Embryology. I have added some material from the other books which can be seen. Now this is the reference source. अब मुझे ख्याल है कि ये आपको ज़रूर पसंद आई होगी, और जहाँ भी आपको मुश्किल हो आपने मुझे कॉल कर लेनी है या मुझे व्हाट्सएप पे पूछ लेना है। डेफिनेटली ये मैं अपलोड करूँगा अपना जो यूट्यूब चैनल है, और उसके साथ साथ ये जो गूगल क्लासरूम है ये उसमें भी अपलोड होगी। तो आप अपनी किताब जो है वो खोल लीजिए, कीथ एल्मूर और लैंगमैन। तो ये मेजॉरिटी मैंने लेक्चर वहाँ से लिया है, और ये मैं रेफ़र इसलिए कर रहा हूँ कि ये आप आसानी से वहाँ से पढ़ सकते हो।